In this second lecture, we're going to look at viral reproduction. We'll see the steps of viral reproduction, both in general and then specifically in bacteriophage. So the viral reproduction will affect the host cell by causing certain cytopathic effects, which means the pathology of the cell. So the biochemical and structural changes in the host cell caused by the virus is replicating itself can lead to several different types of effects. One is lysis, which the cell ruptures and bursts and releases the new viral particles, which of course kills the host cell immediately. Some host cells, upon recognizing they've been infected, will activate some self-destruction genes called apoptosis. We've seen this before. And then there can be widespread effects due to a body's immune system response to try to isolate and lessen the effect of the viral reproduction. In some cases, some viruses will leave the host cell alive temporarily by budding off instead of lysing out of the cell, but eventually this repeated budding will kill the cell. In general, there are 4, maybe 5 steps in a viral infection. The first is that the virus has to recognize the host cell and attach. Then it has to find some way to enter into the cell. Replication means synthesizing the parts and putting the parts of the virus together. And then finally, once they're assembled, the virus has to find a way to get out of the host cell. So this first step, attachment, involves the receptors on the host cell binding to the capsid proteins or the glycoproteins on the virus. These viruses, the receptors have to match very specifically, and therefore the viruses can be very specific in what type of cells that they will infect. After attachment, then the virus has to get in. For bacteriophage, the capsid actually stays outside the cell and injects the DNA into the host cell. We call it naked, without the capsid protection. For eukaryotic cells, the virus actually enters the cell with the capsid and then has to go through a process of uncoding. And then a couple ways to get into the cell would be that the virus induces endocytosis, or in some cases with membrane viruses, they can fuse with the membrane and enter that way. After the viral DNA has entered the cell, then the replication and assembly step begins. Depending on what kind of virus, there can be different strategies. For DNA viruses, the DNA is used to transcribe messenger RNA, which then goes on with the ribosomes to make viral proteins. And then the DNA, because it's a DNA virus, it has to be duplicated to make new viral genomes. With RNA viruses, the RNA makes complementary RNA if necessary, then transcribes that into messenger RNA to make viral proteins, makes copies of the RNA to build into new viral genomes. And then finally, the RNA retroviruses, they come in as RNA and then use reverse transcriptase to make the RNA into DNA, incorporates into the host cell's genome, and then directs the synthesis of new viruses by making messenger RNA from the DNA that's already in the host genome. In order to get out of the cell, to release the viruses, the virus may induce lysis, which would be to kill the host cell and burst the cell, releasing the viruses in the environment. Or the virus could slowly bud off. Usually these are enveloped viruses that take part of the membrane with them and not immediately kill the host cell, but eventually kill the host cell. For influenza viruses, which we have going around right now, the virus will recognize the host cell through the glycoproteins on the surface of the virus and then the proteins on the surface of the host cell. The virus is then engulfed. It then removes the capsid, exposing the nucleic acid to the cell, taking over the cell, begins producing viral parts that then self-assemble into new viruses, and eventually the viruses are released to go invade neighboring cells. For bacteriophage, which are viruses that infect bacteria, and you see in the image the larger structure is the bacterial cell, and all these smaller viruses are attacking it. There are two different types of infectious cycles. There's the lytic cycle, which, as the name sort of implies, results in lysis of the host cell at the end of the cycle. And then there's lysogenic cycle, which has a very unique feature to it. In the lytic cycle, what you see is that the virus is going to infect the host cell, injecting the viral DNA in. The viral DNA goes in and then takes over the cell, making the cell produce more viral particles. Once a certain number of viral particles have been accumulated, then the virus induces the cell to lyse, releasing the viral particles to go out and find a new host cell. For the lysogenic cycle, slightly different. Notice now that the virus is going to inject the DNA in, and the DNA then is not going to immediately take over the cell, but insert itself into the host cell chromosome. Now, every time the host cell replicates itself, it makes a copy of the virus as well. So as the host accumulates, the virus accumulates. And then under stressful conditions, the virus will pop out of the host cell and begin to start a new lytic cycle. So there's a connection between the lysogenic cycle, where the viral genome has inserted it into the bacterial genome and replicates with the bacteria until it pops out. And then it starts to take over the cell like we saw with the lytic cycle, and eventually tries to get itself out of the host cell before the host cell were to die. There are some questions in the textbook called the visual connection. I'd like to go over some of these every once in a while. So which of the following statements is false? So we look here at number A. It says, in the lytic cycle, new phages are produced and released into the environment. And that is true. The lytic cycle ends with the lysis of the host cell and release of the viruses into the environment. Second one, B, the lysogenic cycle, phage DNA is incorporated into the host genome. We see that's true. That's what makes it different. It incorporates into the host genome and then replicates with the host. C, an environmental stressor can cause the phage to initiate the lysogenic cycle, and that's false. Notice the cell is in the lysogenic cycle on the bottom here until some environmental condition causes it to go to the lytic cycle. So it should have been initiate the lytic cycle. And then cell lysis only occurs in the lytic cycle, and that's true. The lytic cycle is the only one that terminates in the lysis of the host cell.